This screencast will cover DNA replication from Chapter 25. Remember that DNA is a polymer constructed of long chains of nucleotides. Only 4 different nucleotides are used in making all DNA. The nucleotides are all alike and they have a single phosphate group attached to the 5-carbon sugar deoxyribose. However, the nucleotides differ in which nitrogenous base is attached to deoxyribose. There are 4 different bases: adenine, guanine, cytosine, and thymine. We'll use A, G, C, and T to represent these nitrogenous bases. DNA from different individuals or different species vary in the sequence in which these 4 bases are arranged. This figure represents a short segment of DNA. This is what we would see in a single-stranded chromosome. I've made each kind of nucleotide a different color. Notice the phosphate groups and the deoxyribose sugars, indicated by P's and D's, make up the sides of our ladder-like DNA molecule. And the nitrogenous bases are paired in the middle, much like the steps of a ladder. Adenine, A, and thymine, T, are always paired together, and also cytosine, C, is always paired with guanine, G. This arrangement is due to the fact that A will only form hydrogen bonds with T, and C will only bond with G. DNA replication must occur before a cell can reproduce, either by mitosis or meiosis. Copies of the DNA are needed so that each new cell will receive an appropriate set of chromosomes. During DNA replication, each single-stranded chromosome makes an exact copy of itself. At this point in time, the copies, called sister chromatids, are attached to the centromere. The entire structure, consisting of 2 sister chromatids, is called a double-stranded chromosome. When I was drawing the stages of mitosis and meiosis, I used X's to represent these double-stranded chromosomes. During mitosis and meiosis, the sister chromatids separated and went to different cells. Let's start DNA replication. Remember, we're currently in the nucleus during interphase. This figure represents a very short segment of a single-stranded chromosome with only 6 base pairs. The human genome, which is the sum total of all the nucleotides found on all 46 chromosomes, contains about 3 billion base pairs. DNA replication begins with an enzyme moving along the length of the chromosome, breaking the hydrogen bonds between the base pairs. This is much like unzipping a zipper. Each half of the original chromosome is now going to be used as a pattern for completing each sister chromatid. This is possible because A will only bond with T and C with G. Individual nucleotides from the cytoplasm are delivered to the nucleus. Wherever there is an A on the DNA, T is brought in. C's are also matched up with G's, and so on. In a stepwise fashion, one at a time, nucleotides are added down the length of each half of the DNA. When DNA replication is completed, we now have 2 identical copies of the DNA. These copies are sister chromatids. At this point in time, they are attached to each other at the centromere. The entire structure is composed of 2 sister chromatids, and it's a double-stranded chromosome. This completes DNA replication.